Imagine the most barren, toxic, industrially destroyed soil on the North American continent. I mean, a landscape that has just been thoroughly cooked by decades of chemical fallout.
Yeah, we are talking about soil so loaded with heavy metals and, so wildly acidic that it essentially functions as an active poison to biological life.
Right. Just looking at it, you would assume nothing could ever grow there again.
Absolutely nothing.
But now I want you to imagine a plant that looks at that absolute wasteland and not only survives, but actually thrives. A plant that actively establishes a foothold in the poison.
It anchors itself in there.
Exactly. It anchors itself and then proceeds to pull invisible nitrogen right out of thin air, ultimately turning that dead rock into a nutritional powerhouse.
It is really the ultimate story of biological resilience.
Yeah.
What you are describing is a phenomenon that forces us to completely rethink how life rebuilds the biosphere from the ground up.
It totally flips the script.
It does. In the botanical community, this plant has earned a reputation as the ultimate badland pioneer because it doesn't just tolerate the destruction. It engineers a completely new ecosystem in places where almost nothing else can survive.
And that is exactly what our mission is for this deep dive today. We are going to act as your personal field botanists, and we are going to explore exactly how this specific, unassuming shrub performs what I can really only describe as ecological alchemy.
Ecological alchemy is a great way to put it.
We are diving into the world of the silver buffaloberry, scientifically known as Shepherdia argentea, and our source material today covers everything from botanical research to Superfund site reclamation data.
Yeah, and we have biochemical analyses too, which get really intense later on.
Oh, they are wild.
Mhm.
But to really understand how this plant works, we have to start by looking at its fundamental biological architecture. Because, when you hear the name silver buffaloberry, it sounds quite delicate.
Very delicate, like a nice little garden plant.
Right. But the reality of encountering one in the wild is very different, isn't it?
Oh, very different. If you stumble across a mature specimen, you are not looking at a delicate little berry bush. You are looking at this erect, perennial, deciduous shrub that can often grow into a small tree.
Like, how tall are we talking?
They routinely stand anywhere from 2 to 6 meters tall. So that is roughly 6 to 20 feet. And they can spread out just as wide, forming these massive, impenetrable thickets.
Wow, 20 feet is huge for a shrub.
Yeah, and they are incredibly long-lived for a shrub, too, often surviving 50 to 60 years in environments that regularly kill other vegetation.
Which is just staggering. And taxonomically, just to ground us, they belong to the order Elignales and the family Elygnacea.
Right, which is commonly known as the oleaster family.
I want to talk about the visual, though, because it is striking. And it actually ties into the history of how this plant was formally introduced to the scientific community.
Yes, the historical record is really fascinating here. The first formal collection of the silver buffaloberry for scientific evaluation was done by Meriwether Lewis.
Of Lewis and Clark.
The very same. This was in 1804 during the Lewis and Clark expedition. He collected the specimen right at the confluence of the Niobrara and Missouri rivers.
Which is in what is now northeastern Nebraska, right?
Exactly.
So you have to picture Meriwether Lewis out in this vast, sweeping prairie landscape. It's an environment characterized by harsh winds, intense sun, extreme temperature fluctuations.
Just a brutal place to be a plant.
And amidst the standard prairie grasses and the usual riparian vegetation, this massive, shimmering silver thicket just stands out.
It would have been totally impossible to miss.
Yeah.
Because the foliage is completely unique. As the young twigs emerge, they are covered in these spiralized structures called stellate hairs.
Stellate meaning star-shaped.
Yeah, stellate means star-shaped. These tiny star-shaped scale-like hairs give the entire young plant this white, almost mealy appearance.
Like it's dusted in flour or something.
Precisely. And then as the twigs age, the bark turns a tough, rugged gray. But the most defining feature, the one Lewis certainly noted, is the leaves.
Right, the leaves are what give it the name.
They are simple, arranged oppositely on the stem, and somewhat oblong, but they are completely covered in dense, waxy, silvery scales on both the top and the bottom sides of the leaf.
And that is a crucial detail for anyone out there listening who might be trying to identify this plant in the field, because there is a very close relative that looks almost identical at first glance.
Yes, its close cousin is Shepherdia canadensis.
Commonly called the Canada buffaloberry.
Right. And the defining taxonomic boundary between the two is found underneath the leaf. If you take a leaf of the Canada buffaloberry and flip it over, the underside is covered in rust-colored scales.
Oh, interesting. So it's like a two-tone effect.
Exactly. But the silver buffaloberry, Shepherdia argentea, is silver through and through, top and bottom.
Okay, so beyond the silver leaves, as these plants mature, the branches do something very aggressive.
They do. The 2 to 3-year-old twigs actually terminate in very sharp, rigid spines.
So you have this massive, sprawling thicket covered in thousands of tiny silver scales, and it is tipped with literal
It's
daggers.
heavily fortified.
I mean, it sounds less like a plant and more like a medieval fortress. I have to ask about the evolutionary logic here. A plant puts a tremendous amount of metabolic energy into growing thorns and synthesizing complex waxes for these scales. Why does it need all this defense?
Well, the spines serve a dual purpose, but the primary function of the entire architectural setup is defense against the climate.
The climate, not animals.
We tend to think of thorns strictly as a defense against herbivores. And, while it's true that the sharp spines help deter browsing animals like deer or cattle from eating the tender young shoots before the plant is established, the real enemy is the environment.
Because the plains are just that harsh.
The Great Plains and the Intermountain West can be incredibly unforgiving. Those silver stellate scales are not there just to look pretty. They are a highly functional reflective shield.
So they are essentially bouncing the sunlight away,
Exactly. They significantly increase the leaf's albedo.
which is its ability to reflect solar radiation.
Right. During the brutal, cloudless summer months on the prairie, a normal green leaf absorbs a massive amount of solar energy, which heats up the leaf tissue.
Like a dark car sitting in a parking lot.
Perfect analogy. And if the leaf gets too hot, the proteins and enzymes responsible for photosynthesis begin to denature and break down.
It cooks itself?
The plant essentially cooks itself, yeah.
Yeah.
But the silver scales of the buffalo berry reflect a large portion of that intense light away, keeping the internal temperature of the leaf much cooler.
Wow. And I imagine the wax plays a role in water conservation, too.
A critical role.
Yeah.
Those scales, combined with a thick, waxy cuticle, dramatically reduce transpiration.
Just to clarify, transpiration is the process where water evaporates from the microscopic pores, or stomata, on the underside of the leaf.
Yeah, you've got it. When you live on harsh prairies, rocky slopes, and windswept badlands where rainfall is scarce and unpredictable, you have to hoard every single drop of moisture your roots manage to find.
You can't afford to sweat it out.
Exactly. The waxy scales create a microboundary layer of still air right next to the leaf's surface, which slows down the rate of evaporation.
So it's basically wearing a windbreaker.
A very shiny windbreaker. This physical armor allows the buffalo berry to survive extreme drought and devastating cold winds that would completely desiccate and kill more delicate vascular plants.
So the plant has essentially built a fortress to survive a perpetual siege by the sun and the wind. And just to round out the basic biology before we get into the really wild stuff, it's also dioecious,
Yes, which means it has completely separate male and female plants.
which is somewhat rare, isn't it?
It is relatively rare. You need a male plant nearby to pollinate a female plant if you want those bright red or orange berries to form.
But as intense as all this above-ground armor is, the research reveals that its above-ground defenses are absolutely nothing compared to its below-ground capabilities.
The above-ground morphology is just the survival mechanism for the climate. The below-ground physiology is what allows it to dominate environments that are chemically toxic.
Which is where this story gets really crazy.
Oh, definitely. If you look at its ecological distribution, you will certainly find it growing happily in moist riparian zones like along the banks of streams and rivers. But its true superpower, the trait that fascinates botanists and ecologists, is its extreme tolerance for soil conditions that represent a death sentence for almost anything else.
We are talking about severe, profound extremes here. The data shows it thrives in incredibly alkaline soils, easily handling a pH ranging from 6.5 all the way up to 8.5.
And sometimes even higher than that.
And it has a remarkable tolerance for saline environments, meaning soils that are heavily burdened with salt.
To understand how extreme that is, we need to look at what high pH and high salinity actually do to a normal plant root. Let's take salinity first.
Okay, let's break it down.
Plant roots absorb water through a process driven by osmosis. Normally, the concentration of solutes like sugars and minerals inside the root cells is higher than the concentration in the surrounding soil.
Because the root is packed with nutrients.
Right. And because water naturally moves toward the higher concentration of solutes, it flows from the soil into the root.
But when the soil is heavily laden with salt, that dynamic flips.
Precisely. The soil becomes hypertonic. The concentration of salt in the soil water is suddenly much higher than the solute concentration inside the plant's roots.
Oh, wow. So the pressure goes the other way.
The osmotic pressure reverses completely. Instead of water flowing into the plant, the salt actually pulls water out of the plant's cells.
That is brutal. So the root physically cannot absorb water no matter how wet the soil is.
It causes severe osmotic stress, and the plant essentially dies of dehydration while sitting in damp dirt.
And then on top of that, you add the alkaline factor, because a pH of 8.5 chemically locks up nutrients, right?
Exactly. Even if there is iron or phosphorus sitting right there in the dirt, the high pH alters the chemical state of those minerals, so the plant's roots just can't absorb them.
The plant starves while surrounded by food.
Yet the buffaloberry navigates both the osmotic stress and the nutrient lockout with ease. And the ultimate undeniable proof of this capability is a specific genetic lineage known as the Mill Creek germplasm.
Yes, this was the part of the research that really hooked me. The Mill Creek germplasm is a specific natural selection of silver buffaloberry, but it wasn't sourced from some pristine national park.
Not at all.
It was found growing on one of the most toxic, industrially altered, and heavily polluted landscapes in all of North America.
The Anaconda Smelter Superfund site in Deer Lodge County, Montana.
The history of this place is just wild.
The history of the Anaconda site is really a grim chapter in industrial ecology. Between 1884 and 1980, this area was the site of massive copper smelting operations.
Almost 100 years of smelting.
Yeah, the Washoe Smelter, which operated there, had one of the largest freestanding brick smokestacks in the entire world. And for nearly a century, that stack, along with earlier operations, was continuously pumping out atmospheric sulfur dioxide and heavy metal emissions.
For almost 100 years, the fallout from those smokestacks was just blanketing the surrounding hillsides.
When you release that much sulfur dioxide into the atmosphere, it mixes with atmospheric moisture and essentially rains down as dilute sulfuric acid.
Like actual acid rain.
Literally acid rain.
Yeah.
And over decades, this relentless acid deposition completely devastated the surrounding soil architecture. By the time the smelter was finally shut down in 1980, the top 18 inches of soil across vast tracts of land had become highly acidic.
The field measurements recorded the surface pH dropping as low as 4.0 to 5.0, which is acidic enough to dissolve heavy metals.
It is.
And that brings up the real horror of the Anaconda site. Because of the region's natural geology, the soil deeper down from 18 inches to 60 inches is strongly alkaline.
It's a chemical nightmare.
So you have a soil profile where the top 1.5 feet is a highly acidic zone, and right below that, it abruptly swings to highly alkaline.
That wildly fluctuating pH profile alone would be enough to prevent almost any native tree or shrub from establishing a deep root system. But the altered pH is only the mechanism that unleashed the real danger.
Because the smelting process didn't just release sulfur.
No, it released thousands of tons of heavy metal particulates. That top layer of acidic soil became laced with phytotoxic levels of arsenic, copper, lead, and zinc.
And when we say phytotoxic, we mean literally, biologically toxic to plant life. How do heavy metals actually kill a plant at the cellular level?
Heavy metals cause acute ion toxicity. Plants require certain trace metals like zinc, copper, and iron to function as cofactors in crucial enzymatic reactions.
They need a little bit of them to survive.
Right, they have specific receptor sites on their cells designed to absorb these necessary ions. The problem is that toxic heavy metals like lead and arsenic often mimic the chemical structure of those essential nutrients.
Oh, so it's a trap.
It's a biological trap. The plant's roots absorb the lead, thinking it's something useful like iron or calcium. Once inside the cells, these toxic heavy metal ions bind to the active sites of enzymes, completely blocking them.
They just gum up the works.
They disrupt the electron transport chain in the mitochondria, shutting down cellular respiration. They interfere with the chlorophyll molecules, immediately halting photosynthesis.
Wow.
They even bind to the nucleic acids, literally shattering the plant's DNA.
So the Anaconda site presents a chemical barrier that is so complex, so acidic, and so toxic that it defies all natural reclamation. If you try to plant a standard vascular plant there, its roots are burned by the acid.
Starved by the alkaline subsoil.
Right. Dehydrated by the disrupted soil structure and chemically poisoned by the heavy metal.
It is an absolute death zone. Yet, researchers surveying the area discovered the Mill Creek selection of the buffaloberry, happily growing right there on what is known as Bee Hill, just south of Anaconda.
It was just living there.
It had established itself directly in soil, heavily loaded with historic mine waste. And when researchers collected seeds from those specific plants and grew them in test orchards to evaluate their genetics, the plants exhibited incredible vigor.
Even without any supplemental irrigation, right?
None. And they quickly grew into massive 10-foot-tall thickets.
Here is where it gets really interesting for you listening. How is it not being poisoned? If the soil water is saturated with toxic ions, how does the buffaloberry pull the water it needs without pulling the poison right along with it?
That is the central mystery, and the answer unlocks the entire ecological mechanism of this species. The silver buffaloberry does not survive the anaconda superfund site alone.
It has a partner.
It manages this seemingly impossible feat by relying on a highly specialized microscopic partner that lives deep inside its root tissue.
We are talking about a phenomenon called actin-or symbiosis, and the mechanics of how this works are just mind-blowing.
The silver buffaloberry is classified as an actin-or plant. This means it has evolved the complex biological machinery necessary to form a symbiotic relationship with specific nitrogen-fixing actinobacteria.
And the bacteria in question belong to the genus Frankia, right?
Yes, the genus Frankia.
Yeah.
To truly grasp how profound this partnership is, we have to look closely at how the plant builds its underground infrastructure to house these bacteria.
Below ground, this plant is an absolute powerhouse.
Right.
It doesn't just send down a few flimsy roots. It drives massive, thick primary taproots straight down into the earth to anchor itself against the wind.
It has just to physically survive the prairie.
Right. And it also sends down deep sinker roots specifically designed to chase the water table during droughts. But the really aggressive part of its architecture is the lateral system.
The lateral roots are incredible.
It shoots out this massive network of spreading rhizomes and creeping lateral roots horizontally through the topsoil.
And those lateral roots are not purely for nutrient gathering. They are highly vegetative. After about 5 years of initial growth, these lateral roots start producing vegetative suckers.
Meaning they sprout new plants?
They literally shoot new stems up through the soil surface, generating genetically identical clone plants.
Wow, so that's how it makes those thickets.
Exactly. Over a few decades, a single individual buffaloberry can expand into a sprawling thicket that binds tons of soil together in a tight, physical web.
That dense physical network provides the structural foundation to stop erosion. But the chemical magic, the actin-or symbiosis, happens at a microscopic level, right where the Frankia bacteria meet the young root tissue.
That is where the actual alchemy takes place.
Now, many people are at least somewhat familiar with legume plants like peas, beans, soybeans, or clover. Legumes are famous for fixing nitrogen, but they use a completely different process, don't they?
Yes, and the contrast is vital for understanding why the buffaloberry is so much tougher. Legumes partner with a different class of bacteria called rhizobia.
Okay, rhizobia for legumes, Frankia for buffaloberry.
Right. And to get the rhizobia inside their roots, legumes rely on a process called root hair infection. It requires a highly delicate, complex chemical dialogue.
How does that dialogue work?
The legume root secretes specific flavonoids into the soil. The rhizobia bacteria detect these flavonoids and respond by releasing their own chemical signals called nod factors.
Like a secret handshake.
Very much so. If the root recognizes the nod factors, it physically deforms a root hair, curling it around the bacteria. The plant then builds an internal tunnel, called an infection thread, to guide the bacteria down into the root cortex.
So if we connect this to the bigger picture, other plants require a delicate knock on the door, a complex chemical signaling process where the root hair has to perfectly deform to let the bacteria in.
It is a very polite process.
Right. Even other tough actin-or trees, like the alder tree, use this delicate root hair infection method, but the buffaloberry does not.
No. Shepherdia argentea utilizes a much more robust, almost aggressive method called intercellular penetration.
The research detailing this infection pathway is meticulously clear, and I love the visual.
The Frankia bacteria do not bother with root hairs. In fact, the roots of the buffaloberry in the zones where infection occurs are essentially devoid of root hairs entirely.
So how do they get in?
Instead, the Frankia hyphae, the microscopic thread-like filaments of the bacteria, penetrate directly between the epidermal cells of the root. They physically wedge themselves through the intercellular spaces of the root cortex.
So the buffaloberry's bacteria just look at the solid wall of root cells and physically squeeze right through the mortar between the breaks. They force their way in. Why is this brute force entry method evolutionarily superior for a plant living in a wasteland?
What's fascinating here is that the delicate chemical handshake required by legumes and alders is incredibly fragile. The flavonoid signals and the nod factors are complex organic molecules.
And I'm assuming those molecules don't hold up well in a Superfund site.
They are easily scrambled, degraded, or completely destroyed by environmental stressors. If the soil has an extreme alkaline pH, high salinity, or heavy metal toxicity, exactly the conditions at the anaconda site, the chemical signals break down before they ever reach the bacteria.
The knock on the door gets lost in the chemical noise.
Exactly. The root never recognizes the bacteria, the infection thread never forms, the plant never gets its nitrogen, and it starves to death.
But because the Frankia bacteria and the buffaloberry just physically push their way through the intercellular spaces, they completely bypass the need for that fragile signaling cascade.
They bypass it entirely.
Hmm.
The intercellular penetration pathway is far less susceptible to environmental disruption. And once the Frankia hyphae are inside the root tissue, they skip yet another step that other plants require.
Oh, really?
Yeah, in root hair-infected plants, the root cortex has to rapidly build a temporary housing structure called a pre-nodule to contain the bacteria, while the actual permanent root nodule is slowly constructed.
Like a temporary construction trailer.
Exactly. But the buffaloberry skips the pre-nodule entirely. The invading Frankia hyphae grow directly toward the dividing cells of the emerging nodule lobe primordia, the actual construction site of the permanent nodule.
They march straight to the final destination and immediately begin establishing the symbiotic factory.
And the physical factory they build together is an engineering marvel. These nodules aren't just little bumps. They are highly modified lateral roots, but they lack root caps.
And inside the cells of these nodules, the plant and the bacteria co-construct something called a multilaminated lipid envelope to house the Frankia. Why do they need to wrap the bacteria in layers of lipids or fats?
That lipid envelope is the key to the entire operation. The ultimate goal of this symbiosis is to produce an enzyme called nitrogenase.
Nitrogenase.
Nitrogenase is the biological catalyst that takes inert nitrogen gas N2 from the air pockets in the soil and breaks the incredibly strong triple bond holding the two nitrogen atoms together.
It converts that gas into ammonia, which is a bioavailable form of nitrogen the plant can use to build amino acids and proteins.
Right. But breaking a nitrogen triple bond requires a massive amount of energy, which the plant supplies in the form of sugars from photosynthesis.
Yes, but there is a major chemical vulnerability here, isn't there?
A huge one. The nitrogenase enzyme is violently sensitive to oxygen. If oxygen molecules come into contact with nitrogenase, they permanently oxidize and destroy the enzyme. It is completely deactivated.
But roots are growing in soil, and they need oxygen for normal cellular respiration.
So you have a paradox. The bacteria need to run an oxygen-sensitive enzyme inside a root that is surrounded by oxygen. The multilaminated lipid envelope solves this.
The layers of fat protect it?
The layers of lipids create a physical, highly regulated barrier that strictly limits the diffusion of oxygen into the bacterial cell, keeping the internal environment hypoxic enough for the nitrogenase to function.
While still allowing just enough oxygen through for the bacteria to actually survive.
Exactly. It's a masterpiece of biochemical engineering.
It really is. It's a highly protected, microscopic underground factory, turning thin air into premium fertilizer built in the middle of a toxic wasteland. But this incredible superpower comes with a very bizarre, highly counterintuitive catch.
It does.
And it's a catch that often trips up humans when we try to cultivate this plant.
It's a phenomenon that researchers often refer to as the Goldilocks paradox of nitrogen fertilization.
Let's look closely at the studies done on a specific cultivated variety of the buffaloberry. The researchers were looking at a hybrid called Shepherdia echihensis tori.
Right. This specific hybrid was created by crossing our native silver buffaloberry with another species, the roundleaf buffaloberry.
To create a sterile, robust plant specifically suited for urban landscaping and drought-resistant gardens. The researchers wanted to determine the optimal nursery protocols. Essentially, how do you grow the biggest, healthiest buffaloberry in a commercial greenhouse setting?
And, the standard operating procedure in the commercial nursery industry is to rely heavily on chemical inputs. If you want rapid, vigorous vegetative growth so you can quickly bring a beautiful, bushy plant to market, you pump the potting soil full of mineral nitrogen.
You use high doses of controlled-release granular fertilizers, or you irrigate with a heavy solution of ammonium nitrate.
Because that's what usually works.
It makes sense on paper. You have a plant that needs a lot of nitrogen to build its massive root system and its dense foliage, so you give it what it wants. But when the researchers applied this standard nursery protocol to the buffalo berry, the entire symbiotic system crashed.
It did. The studies revealed that this plant is hypersensitive to the presence of environmental nitrogen.
How sensitive are we talking?
When the researchers applied a relatively standard dose of just 2.9 grams per liter of a controlled-release fertilizer, or irrigated with a 2 millimolar solution of ammonium nitrate, the results were drastic. It completely and immediately halted nodule initiation.
The intracellular penetration by the Frankia hyphae just stopped.
Stopped cold. 0 root nodules formed. The plant completely rejected the symbiotic partnership.
So, what does this all mean for someone trying to grow this in a nursery? By trying to baby the plant with premium fertilizer, we are actually breaking its biological engine.
Exactly. Less is more. It comes down to cellular economics and the strict conservation of metabolic energy. We mentioned earlier that breaking the triple bond of nitrogen gas requires massive energy.
The plant is paying for this process.
Yes. The plant has to photosynthesize sunlight into sugars, transport those sugars all the way down into the root system, and feed them to the Frankia bacteria to power the nitrogenase enzyme.
Furthermore, constructing the physical nodules and the complex lipid envelopes requires a huge investment of carbon and structural resources.
It is essentially paying a massive biological tax to keep this bacterial workforce employed. The plant is constantly running a highly sensitive internal cost-benefit analysis.
Its roots are basically tasting the soil chemistry.
Right. If those roots detect an abundance of free, easily available mineral nitrogen in the soil, like when a well-meaning human dumps premium synthetic fertilizer onto it, the plant's internal calculus shifts.
It realizes, why should I spend vast amounts of my hard-earned photosynthetic sugar to build a factory and feed these bacteria when the final product is already sitting right here in the dirt for free?
It actively suppresses the Frankia infection because supporting the bacteria would suddenly be a waste of metabolic energy. It downregulates the entire symbiotic pathway.
So it only forms nodules when it's desperate.
The research proved that the plant only actively signals for and permits the Frankia partnership under harsh, nutrient-limiting conditions. The scientists found that optimal explosive nodule development occurred when the plants were grown in pure, low organic matter substrates.
They literally use pure perlite, right?
Yeah, pure perlite, a volcanic glass that contains absolutely 0 nutrients. They grew the buffalo berry in sterile glass, irrigated it with a completely nitrogen-free water solution, and maintained a relatively alkaline pH of 7.5.
And under those brutally stark conditions, the plant's biology went into overdrive.
Recruiting every Frankia bacterium it could find, and building massive, complex clusters of root nodules.
So by withholding fertilizer, nurseries can force the plant to develop its nodules, resulting in healthier plants.
Yes. Once those nodules are fully established, the plant becomes completely self-sufficient. And there is a massive environmental benefit to this approach that goes beyond just growing a tougher plant.
The nitrate runoff, right?
The environmental implications for the nursery industry are huge. In typical commercial nurseries, the heavy reliance on constant nitrogen fertilization leads to significant nitrate leaching. The water runs through the pots, carries the excess synthetic nitrate out of the nursery, and dumps it into local groundwater systems and streams.
Which causes devastating algal blooms and water pollution.
But by understanding and utilizing the buffalo berry's natural actor-hyzal symbiosis, nurseries can produce healthier, faster-growing plants with virtually 0 nitrate runoff. You let the ancient biology do the heavy lifting, rather than relying on modern polluting chemistry.
You have to let the plant struggle just enough so it turns on the machinery that makes it invincible. It is a brilliant evolutionary strategy. Now, this poses a really interesting physiological question. If this plant has developed this massive underground factory, pulling limitless nitrogen out of thin air, and it's surviving on barren rocks and toxic mining waste, it is generating a tremendous amount of synthesized energy. Where does all that energy go?
Once the plant has established its vegetative structure, it directs an astonishing amount of that metabolically expensive energy directly into its reproductive cycle, specifically into the complex chemical composition of its fruit.
And the biochemical analysis of these fruits is where we see just how extraordinary this plant's internal manufacturing capabilities really are.
Before we dive into the specific data tables, it is important to clarify our perspective. We are analyzing these chemical concentrations strictly from a botanical and physiological standpoint. We are marveling at the metabolic extremes this plant achieves in a harsh environment. We are not providing dietary or health advice.
Absolutely. We're just looking at what the plant can do. The data speaks volumes purely in terms of biological capability. Let's look at the specific phytochemical parameters the researchers measured. In late summer and autumn, the mature female plants produce these incredibly dense clusters of small, round berries, which are technically classified as drupes.
They are quite small, usually only measuring about 4 to 10 millimeters in diameter.
But they ripen into this brilliant, fiery crimson red or bright glowing orange. Visually, the contrast of these blazing red clusters set against the silvery, waxy leaves is stunning.
But the liquid chromatography of what is actually inside these tiny drupes is mind-boggling. We are looking at a fruit with an exceptionally high density of specialized phytochemicals and antioxidants.
The most prominent feature of the red fruit, the compound responsible for that intense crimson color, is its immense concentration of carotenoids, specifically lycopene.
Lycopene is a well-known pigment molecule, but the levels found in the silver buffaloberry are staggering.
Okay, let's unpack this lycopene data. I had to read the chromatography table 3 times to make sure I wasn't misinterpreting the units. Most of us associate lycopene with tomatoes. It's the classic example. A standard field-grown red tomato contains roughly 3 milligrams of lycopene per 100 grams of fresh fruit weight.
Right, 3 milligrams.
The analytical tests on the silver buffaloberry measured the lycopene concentration at approximately 27 milligrams per 100 grams of fresh weight. It has 9 times the lycopene of a tomato. It's a botanical multivitamin factory powered by literal badlands.
It is an incredible biological accumulation. But what is truly fascinating from a plant physiology perspective is understanding why the lycopene concentration is so astronomically high.
Right, because it's not trying to build a superfruit for us to eat.
No, it is actually the result of a specific metabolic limitation, a missing step in the plant's biochemical assembly line.
Explain how the carotenoid assembly line normally works in a plant.
In the standard carotenoid biosynthesis pathway, plants synthesize a colorless compound called phytoin. Through a series of enzymatic desaturations, that phytoin is converted into lycopene, which is red.
Okay, so phytone to lycopene.
Now, in most plants, the process doesn't stop there. They utilize downstream enzymes, specifically lycopene cyclases, to convert that red lycopene into other crucial carotenoids, like beta-carotene, which is orange.
And beta-carotene is vital for photosynthesis and photoprotection.
Exactly. But the genetic analysis of the silver buffaloberry reveals that its fruit tissue completely lacks these specific cyclization enzymes.
So the assembly line reaches the lycopene stage, and the conveyor belt simply stops running.
Because it lacks the lycopene cyclases in the fruit plastids, it simply cannot convert the lycopene into beta-carotene or any other downstream product. So as the drupe matures over the summer, the plant continues to pump precursors into the pathway, and the lycopene just endlessly backs up and hoards inside the cells.
It's just a bottleneck.
This metabolic roadblock is what leads to the massive chemical concentration and the intense, almost unnatural red coloration of the berries.
But the liquid chromatography mass spectral analysis found that lycopene is only half of the carotenoid story. The researchers identified another, highly unusual compound that actually makes up the majority of the antioxidant profile.
Yes, the spectral analysis isolated a compound abbreviated as Ma6L. The full chemical name is methyl epo6-lycopenoate.
The data states that this single specific compound represents an astonishing 55% of the total carotenoid content in the buffaloberry fruit. And what blew my mind is that the researchers noted this compound is not detected in common agricultural crops.
You don't find it in apples or strawberries or grapes.
What exactly is Ma6L?
Ma6L is what biochemists call an apocarotenoid ester. Apocarotenoids are typically formed when a primary carotenoid, like lycopene, is enzymatically cleaved or chopped up into smaller pieces.
Like breaking down a larger molecule?
Usually, apocarotenoids act as transient volatile signals, like the compounds that give flowers their scent, or as internal plant hormones, like abscisic acid, which regulates dormancy.
So they aren't usually just sitting around in bulk.
Exactly. It is botanically highly unusual to find an apocarotenoid ester accumulating in massive quantities inside a fruit tissue. The buffaloberry is hoarding all this lycopene because of the blocked assembly line, and then a specific cleavage enzyme is coming along and chopping the lycopene molecules into this unique, highly specific ester.
The metabolic uniqueness doesn't stop with the carotenoid pigments either. The researchers also measured the phenolic profile of the fruit, and the numbers are just as extreme. The fruit contains an immense concentration of complex compounds called proanthocyanidins.
Proanthocyanidins are a class of polyphenols.
Yeah.
In simpler terms, they are condensed tannins.
Tannins, right.
They are large, complex oligomeric structures that plants synthesize primarily for defense, but they also possess incredibly powerful radical scavenging capacities, meaning they act as potent antioxidants within the tissue.
The quantitative measurements came in at an astonishing 505 milligrams of proanthocyanidins per 100 grams of fresh fruit.
That is a massive dose of tannins.
To give the listener a point of comparison, the researchers mapped that against two other famous cold climate superfruits, the sea buckthorn and the chokecherry. The silver buffaloberry has 10 times the proanthocyanidin concentration of the sea buckthorn. 10 times.
And when you look at the total phenolic content, measuring all the various phenolic acids and flavonoids combined, it averaged an incredible 9.06 grams of gallic acid equivalents per kilogram of fruit.
And just to top it off, it also pumps out a massive amount of vitamin C, roughly 209 milligrams per 100 grams. So if we synthesize all this data, we have this tiny 6-millimeter berry growing in toxic dirt, and it is just bursting at the seams with lycopene, unique apocarotenoids, massive loads of tannins, and vitamin C.
And sugar. Don't forget the sugar.
Right. The chemical breakdown shows that it is packed with simple sugars. Fructose and glucose make up nearly 60% of the total sugar content. With all that simple sugar, you would logically assume this berry tastes incredibly sweet, like a tiny wild candy.
You would assume that, but the underlying chemistry dictates a very different sensory experience. Despite the high concentration of fructose and glucose, the fruit is famously intensely tart and highly astringent.
If you were to bite into one, it would not be a sweet experience. Why does it taste so harsh if it has so much sugar?
The taste is driven by two overwhelming chemical factors that mask the sugar. First, that massive load of proanthocyanidins, the condensed tannins, creates a powerful astringency.
Like drinking over steep tea.
Precisely. Tannins physically bind to the proteins in your saliva, causing them to precipitate out. This immediately creates that dry, rough, puckering sensation in your mouth.
And the second factor?
Second, the fruit has a phenomenally low pH. The researchers measured the internal pH of the juice at 3.01. Furthermore, it has a very high total titratable acidity, meaning the cellular vacuoles are just loaded with free organic acids.
The sheer volume of acid and tannins completely overpowers the taste of the fructose. From an evolutionary standpoint, why would a plant put so much energy into making a fruit, packing it with sugar, but then making it so sour and astringent that many animals won't eat it?
It is a highly targeted evolutionary defense mechanism. The buffaloberry does not want its fruit consumed by just anything, and it especially does not want its fruit consumed by microscopic pathogens.
Oh, to stop it from rotting.
That highly acidic pH of 3.01, combined with a dense wall of phenolic tannins, creates an internal environment that severely restricts microbial colonization. It physically prevents ambient fungi, yeasts, and bacteria from breaking down the cell walls and rotting the fruit.
It's essentially acting as a chemical preservative system.
Precisely. The buffaloberry relies on specific wildlife, primarily certain bird species and bears for seed dispersal. By making the fruit highly acidic, astringent, and rot-resistant, it preserves the structural integrity of the drupe on the branch late into the fall, and often deep into the freezing winter.
The fruit remains viable and available on the landscape when all other food sources have vanished.
Exactly, ensuring that its target dispersers will eventually consume it and carry the seeds to new locations.
It is absolute biochemical brilliance. It uses the nitrogen it pulled from the thin air, powered by the Frankia bacteria in its roots, to build an impenetrable chemical preservative system so its seeds can survive the winter storms.
It's a closed loop of survival.
We have gone incredibly micro here, down to the exact milligrams of apocarotenoids and the intercellular pathways of the bacteria. Let's zoom out. Let's move from the microscopic chemistry of a single fruit back to the massive landscape scale.
Because when you put thousands of these highly armed, nitrogen-fixing, heavy metal-surviving super shrubs onto a ruined landscape like the Anaconda site, it fundamentally alters the physical world around it.
This changes how we think about ecological restoration. We usually think we have to bulldoze bad topsoil and truck in chemicals, but this suggests we just need to plant the right partnership.
This brings us to the broader concept of ecological engineering and the true systemic value of the silver buffaloberry in the context of environmental reclamation. In ecology, we classify this plant as a pioneer species.
A pioneer species is the very first hardy organism to colonize a completely disrupted, barren, or severely damaged ecosystem, right?
Yes, but the research makes clear that the buffaloberry doesn't just passively colonize the badlands, it actively terraforms them.
Let's walk through the timeline of how it rebuilds a dead biosphere starting from the ground down.
Step one of terraforming is physical stabilization. If you have highly erodible badlands or slopes that have been completely destabilized by open-pit mining or smelting fallout, the soil structure is gone.
Every time it rains, the soil washes away. Every time the wind blows, it creates toxic dust storms.
When a buffaloberry seed germinates in that waste, those primary tap roots drive deep, and the creeping rhizomes spread wide. Those roots act exactly like physical rebar in concrete. They lock the shifting, degraded soil in place, immediately halting wind and water erosion.
That structural integrity is the required foundation for any future ecosystem, so it physically stops the bleeding of the landscape. What is step two?
Step two is chemical enrichment. Remember the Frankia bacteria housed inside those protective lipid envelopes in the root nodules?
The nitrogen factories.
They're continuously, endlessly pulling nitrogen gas from the atmosphere and converting it into bioavailable ammonia. And crucially, they aren't just feeding the individual buffaloberry plant. That massive biological nitrogen fixation continuously infuses the surrounding rhizosphere, the microlayer of soil immediately in contact with the vast root network, with rich, bioavailable nitrogen.
It basically leaks fertilizer into the surrounding dirt.
Yes, and it also deposits it from above. Every autumn, the buffaloberry shed thousands of its silvery leaves. Underground microscopic fine roots constantly die off and decompose. All this nitrogen-rich organic matter is deposited directly back into the barren mineral soil.
Slowly, year after year, this accumulating organic matter initiates the natural process of topsoil formation in a place where topsoil had been completely destroyed.
It's literally printing fertile topsoil out of thin air and sunlight. But we have to address the elephant in the room regarding the toxic sites. What about the poison?
The heavy metal.
Right. Adding nitrogen and organic matter is wonderful, but if the soil profile is still laced with phytotoxic levels of arsenic, copper, and lead, no other delicate native plants are ever going to be able to use that new topsoil. The heavy metals will just kill them.
This is perhaps the most profound and critical role of the actinorhizal symbiosis. The research highlights a phenomenal capability of the Frankia bacteria themselves.
The bacteria handle the heavy metals. Specific strains of heavy metal-resistant Frankia don't just passively survive the toxicity of a site like Anaconda, they actively manage and neutralize it. Detailed studies have shown that these specific Frankia strains have the biochemical ability to bind toxic heavy metal ions, such as lead, directly to their cellular surfaces within the root zone.
I want to make sure I grasp the mechanics of this. The bacteria physically grab the heavy metal ions out of the soil water and hold on to them.
Yes, it is a process called localized biosorption or immobilization. The cell walls of the Frankia bacteria contain specific functional groups that chemically bind with the heavy metal cations.
So the bacteria act as a microscopic subterranean filtration system.
By binding the heavy metals directly in the rhizosphere, they lock the toxic ions in place.
Which means the lead and the arsenic are no longer floating free in the soil water.
Exactly. And because the metals are immobilized by the bacteria on the exterior of their roots, it prevents those toxic ions from being taken up into the plant's vascular system.
The poison is blocked from traveling up the xylem of the stem, and therefore it is never deposited into the leaves or the fruit.
That is a massive ecological safeguard.
This completely changes how we approach the concept of environmental restoration. When we look at a destroyed Superfund site, the traditional human impulse is industrial. We think we have to bring in bulldozers, scrape away millions of tons of poisoned topsoil, haul it away in massive dump trucks, and then truck in millions of dollars of artificial chemical fertilizers and imported clean dirt to force the landscape back to life.
But the biology of the buffaloberry suggests an entirely different path. If we connect this to the bigger picture, it requires a complete paradigm shift in environmental management.
Because degraded, marginal, or even highly toxic landscapes don't always require expensive, high-input, brute force artificial interventions.
In fact, as we learned from the Goldilocks paradox, adding artificial chemical interventions like synthetic nitrogen fertilizer actively breaks the natural healing mechanism.
Right, because if a restoration crew helicopters in and sprays synthetic fertilizer all over the Superfund site to try and speed up the growth, the buffaloberry's internal cost-benefit analysis kicks in.
It senses the free nitrogen, It shuts down the Frankia infection, the nitrogen-fixing factory closes, and most importantly, the heavy metal filtration system stops working.
We have to trust the biology and get out of the way.
When allowed to function naturally, the buffaloberry acts as the master architect for a brand new, localized ecosystem. The physical root web stops the erosion. The constant shedding of nitrogen-rich leaves and roots gradually alters the soil microclimate, increasing its fertility and its capacity to hold moisture.
And the localized immobilization of heavy metals by the Frankia ensures that the surrounding environment is rendered safe.
This protects the entire food chain. The birds that eat the berries, the deer that browse the leaves, the bears that forage in the thickets, from acute heavy metal contamination.
It creates a safe, fertile oasis in the middle of a toxic desert.
And over time, decades, perhaps a century, this localized terraforming dramatically alters the trajectory of the landscape. The improved soil and the protective shaded canopy of the silver buffaloberry thicket create a microenvironment that finally allows for the natural recruitment and establishment of late successional plant species.
The delicate native grasses, the complex forbs, and eventually other non-nitrogen-fixing deciduous trees can finally take root and survive in the exact same soil that would have killed them decades earlier.
It literally sacrifices itself to build a safe nursery for the rest of the forest out of its own cast-off leaves and bacterial partners. It is an incredibly profound biological process.
As we wrap up this deep dive into the source material, I want to distill everything we've discussed into a clear, shareable takeaway for you listening. If there is one core ecological lesson to pull from the hundreds of pages of botanical research, chromatography data, and reclamation studies we've explored today, it is this. Soil degradation is not fundamentally a chemistry problem that requires a massive artificial chemical solution.
It really isn't.
True ecological healing cannot be achieved by fighting the harsh, toxic reality of a destroyed landscape with brute force human inputs. Instead, we must engineer a biological framework. We have to utilize the natural, microscopic, intricately co-evolved plant microbe symbioses that already exist.
The tools are already there.
The Earth already possesses the advanced technology required to heal its most grievous wounds. We simply have to deploy the right native partnerships and have the humility to let them work.
It is a lesson in patience and biological respect. And it leads to a final, provocative thought that I want to leave you with. Something for you to deeply mull over the next time you are out on a hike, driving past an abandoned industrial lot, or looking at an overgrown patch of weeds on the margin of a highway.
What should they be looking for?
Think about this. If the silver buffaloberry, a thorny, dense, aggressively thicket-forming scrubby bush that most people might dismissively hack down as an unkept nuisance, is secretly a high-tech biochemical factory capable of terraforming our most toxic industrial scars. What other overlooked, weed-like native plants are currently sitting on the margins of our landscapes?
Waiting to be understood.
Exactly. What other invisible microscopic symbioses are quietly holding the biological keys to restoring the future of our biosphere, just waiting for us to finally pay attention?
The answers are out there, usually hidden right beneath our feet in the dirt. Thank you for joining us on this deep dive. We loved acting as your field botanist today, and we hope you never look at a thorny prairie shrub the same way again.